However, this current lecture and the following one are going to have less audience participation than the others. And I want to make sure everybody can see the slide deck, all right? Give people just one more minute to get online. Okay. Everyone all right? Okay. So what I'd like to talk about today, and some people may have already seen this talk given elsewhere, I'd like to talk about today is some of the problems that you can solve using CompuCell 3D. As I mentioned, most of this workshop is going to use the example of modeling viral infection and immune response. But CompuCell is a very flexible tool, can be used for all sorts of other applications. And this lecture will primarily review those other applications. So we're going to cover a little bit of a background about why we need to do this kind of modeling in the first place, what virtual tissue modeling is, and then I'm going to give very brief examples of some of the problems that people have used CompuCell to address over the years. And then at the very end, I'm going to have a few-minute exercise in which I'd like you to think about the biological problem that you're interested in and begin to think about how you might organize that problem in a way that could be solved with CompuCell. That's actually a long exercise. We're not going to do all of it now, but I would like you to think about it freshly in the context of the things that I'm going to present, and then have people come back to it essentially as a homework problem or as a long-term research problem. So we're going to be addressing biomedical problems in general. Those could be problems in developmental biology. How do you organize tissues? They could be problems in homeostasis. How do you maintain a tissue once you've built it? They could be problems of failure of homeostasis, of which cancer is the most classic example. We're going to talk about infectious disease modeling. Another area that CompuCell has been used for extensively is in modeling toxicology, how chemical compounds can damage cells and tissues. And people have also used CompuCell-like approaches in CompuCell to model things like tissue engineering. In all of these problems, we have interactions at multiple scales, and there are complex interactions not only between scales, but between biochemical and physical processes, mechanical processes, electrical processes, and so on. And so we have to understand how those interactions work. Why do we need quantitative modeling at all? Well, I suppose people believe that there's some interest in it or they wouldn't be here today. But in general, most experimental biology, classically, bioinformatics, tells us what components are and how they connect. But for example, if I have a feed-forward circuit with incoherence, which I'm sure Herbert talked about last week if you were on Herbert's class, A promotes B and C, B promotes D, but C inhibits D. From the diagram itself, you can't tell whether D increases or decreases when you increase A. When you have a cell moving on a substrate, does the cell move faster or slower as the substrate stiffness increases? Or if you have a tumor that's interacting with the vasculature, how does the vascular interaction change the shape of the tumor? Those are all questions that you can't answer just by looking at a set of diagrams or relationships. You actually have to do a quantitative simulation. Those quantitative simulations come under a variety of names, depending on where you're coming from. We often call them simply multi-scale models. They're often called mechanistic simulations. Sometimes they're called virtual tissues. And in the engineering world, those kind of virtual simulations are used, mechanistic virtual simulations are used to design almost everything. But mechanistic simulations in general are fairly new in biology and biomedicine, with some specific exceptions, which I'm going to talk about. But it's an area that's growing rapidly. And so tools like CompuCell are designed to make that easier. One of the issues that we're going to face when we deal with biology is that the range of length scales is very large, And depending on the questions you're asking, and I want you to focus today in this talk on the questions you're interested in. The questions you're going to ask, there may be different modeling approaches and different things that you need to think about. So if I'm interested, for example, in doing radiation therapy on the brain for glioma, glioblastoma, I'm modeling volumes of cubic centimeters. And when I'm going to do that, I'm going to use what are called finite element packages, solids modeling packages. I'm not going to be able to model the behavior of individual cells. That would also be true for orthopedic implants or for models of the heart for designing heart surgery. On the other hand, if I'm going to do rational drug design, I probably need the individual molecules, and it may be the individual atoms in the molecules, to know what's happening at a molecular level. If I'm modeling individual atoms, I probably can't even get up to the scale of a single cell, let alone to a tissue. Network models, which is what Herbert covered last week, are non-spatial models that talk about the relationship between components. For example, that A promotes B and C, and B promotes D, and C inhibits D. That's a network model. There are also pharmacokinetic models, which describe how chemicals are transported and transformed in the body. Those are all non-spatial models, but they can be very powerful, and they're used widely in industry. What we're going to be talking about this week are what are called multicellular models, where we have explicit representations of the individual cells. And those are particularly useful when things happen at the individual cell level. For example, a mutation in a tumor always happens initially in a single cell, or when cells are moving relative to each other. In many tissues, cells move a lot relative to each other, especially during embryonic development and wound healing. There are other tissues in which there's relatively little relative cell movement. So the heart contracts a lot, but the cardiomyocytes don't move relative to each other when the heart contracts. And so heart modeling typically doesn't involve single cell modeling. So the virtual tissues that we're going to be talking about are going to be multi-scale models. Typically, in our models, we're going to include a layer of control at the molecular level, which will be a network model. That will control cells and what those cells do, so a multicellular model. And then there'll be a larger scale model of things like chemical fields in the tissue, and that will also interact with the other two layers. Sometimes we'll also have a highest level in which we have a representation of transport in the body, a PBPK model, which is also an ODE model, also a network model. Depending on what we're doing, the timescales could range from fractions of a second to years. Typically in the simulations we're going to run, the smallest timescales we're going to look at will be minutes, and we'll be going up to days and years in terms of the length of the simulation. These are mechanistic models. They're agent-based. That is, we have a representation of each component, that would be a cell in the model, and they're multi-scale. And what we're going to be looking at in particular is patterning. We've seen patterning occur already in the simulations we've run, the organization of endothelial cells into capillaries. We've seen the organization of dividing cells in the colonic crypt into the pattern of cells in the colonic crypt. Those are patterning problems. We're going to be focusing on mechanism, which makes this very different from bioinformatics. They're additive. Models start with nothing, and nothing exists in a model unless you explicitly add it. We're going to focus on scales from cells to organisms, and we're going to be based on models of how components work. How do viruses replicate? How do cells move? How do cells grow? How do they die? And our outputs will be time series, which are more comparable to biology, and then movies, which are comparable to what you get when you look, for example, through a 20-power microscope. If we look at real experimental developmental problems, we see that they're very complex, and they also involve a lot of scales. In the top, we have branching morphogenesis in an explant of lung tissue. On the right-hand side at the top, we have the development of a zebrafish embryo. Every cell in that zebrafish embryo was labeled by a team and being tracked. At the bottom, we see blood vessels growing also in the zebrafish as the zebrafish is developing. And on the bottom right, we see the development of early stages of a chicken embryo. All of these contain aspects of problems that we've addressed or others have addressed using CompuCell or programs like CompuCell. Building virtual tissues is not easy. There's a lot of missing science. And so we have to be humble when we build virtual tissue models. There are many components in a given simulation. Typically, we can't model as many cells as there really are in a simulation. There are a lot of different kinds of cells, and you could have two cells that are both of a given type, two cells that are epithelial, but that behave differently. It's not like engineering where every screw of a particular type is the same. In this case, two cells could be nominally the same, but actually behaviorally different. We don't have the ability to describe things in complete detail. In fact, choosing what to leave out is one of the important things that we do when we build models. And in many cases, comparing experiment and simulations is not so easy. How do we compare two movies to ask, has our simulation movie reproduced what we see in experiment? Biology is stochastic and very noisy. So each time we run a simulation, even with identical parameters, we'll get slightly different results. How do we interpret that? These are all things that we have to think about when we frame a problem that we want to solve. In some cases, we may not know all of the basic processes and interactions that are occurring. And often modeling will throw up our ignorance and help us understand what experiments we need to do to really understand how a biological problem works. Coupling between elements is close, it's tight, and that makes things a little more complicated to model numerically. And the range of length and timescales is very large. Again, we have no way of getting anywhere near the idea of building what's called an ab initio model, where you imagine modeling every atom in a cell and every cell in an organism. It's too much out of it. If we come to clinical applications, there are what we would call virtual tissue models that are used clinically. Typically, these are continuum virtual tissue models, that is, models that don't include individual cells. Places where they're used a lot would be the design of orthopedic implants. If you have a knee replacement, that's often designed using a mechanics virtual tissue model. There are non-spatial models, what are called virtual twins, which do data mining to try to predict how your health will change in time. There are molecular dynamics studies of individual molecules, which I mentioned for rational drug design. Some kinds of heart surgery are now designed with personalized virtual tissue models. The team, for example, Andy McCullough at University of California, San Diego, does very beautiful modeling that's used in heart surgery design. Cancer radiation is used modeling, mechanistic modeling for a long time to optimize dosing schedules. And also for surgical protocols. So the key thing when you're going to build a model is to think about what scales and approaches are appropriate. And that's going to depend on the questions that you want to answer. In general, you want to build a model that's the simplest to answer the questions that you're focusing on. You may not know the answer to what's the most appropriate or simplest until you try it. But if you can build a non-spatial model of a phenomenon, for example, a network model of infection, and that works well enough, you don't necessarily need to do the kind of spatial modeling we're going to talk about this week. And in the case of viral infection, there's been a lot of very successful work done on modeling in non-spatial contexts. People like Alan Perlson have done very, very useful clinical work modeling viral infections without going to spatial structured models. So when you come to dealing with spatially structured models, you want to ask the question, why do you need to build a spatial model? What is it about the spatial organization of the system that makes that spatiality really important? Ideally, you want to use the simplest model possible. If fewer parameters, it's run faster, they're easier to fit. And one of the things we're going to focus on in this class, which is what's new about it, is starting from ODE models, starting from network models of viral infection. We're going to build spatial models where the spatial model inherits the parameters from the ODE model. That means you have the speed and efficiency of model fitting and parameter identification from the ODE models, but you have the flexibility and the power of spatial modeling. And during the next couple of days, we're going to focus on how you take ordinary differential equation models and translate them into spatial models in a consistent way. There are many different kinds of multicellular model. CompuCell uses something called GGH, or cellular POTS model, CPM. There are everything from cellular automaton models to flock models to center models, which are very powerful because they allow you to do many, many cells, but don't have spatial resolution of the cells. The cells don't have shape. What are called vertex models, which are very often used in modeling tissue folding and bending in early development. Multi-element models, which combine the center model approach with some finite element techniques, and a variety of others. Finite element models are classic solids models that people like Andy Samoji here at IU are developing. Each one of these methods computationally has advantages and disadvantages in terms of number of cells, speed, detail, and so on. They're all valuable in their way for different problems. Just to show you a little bit of a zoo of some of the pictures of different modeling methodologies, these are all center model, these are all point-like models, or cellular automaton models. So in this case, each cell in each one of these simulations is a point, doesn't have any spatial detail. In this case, we're going to the other extreme in that upper left corner. They're very, very detailed models, which are triangulated. These are basically finite element solids models, fluid models of blood cells flowing in a capillary. In this case, there's a huge amount of detail, but those are very, very expensive computer simulations to run. So running 1 second of simulation time means hours and hours and hours of supercomputer time. In the middle, we've already seen our little vascular tumor simulation, which we'll come back to later in the course probably, and then other examples of different kinds of modeling. The bottom right shows what are called vertex models, which again are used typically for folding, modeling buckling transitions in epithelial sheets. There are also a variety of simulation software packages for implementing these. These are some of the packages that simulate center models. There are many of those, really lots of center model packages. And there are also a variety of packages for doing simulations of cells with explicit shapes. In this course, we're going to be talking about CompuCell3D, but there are others. And those are always interesting to explore. The Morpheus package from Technical University of Dresden, to me, is particularly interesting. It uses the same methodologies that we do. And has a very beautiful way of specifying models. So if you like what you see here and you want to explore more, I certainly encourage you to take a look at Morpheus. Unfortunately, there isn't a good textbook available about this kind of modeling. The closest thing we have is this book from several years ago by Scianna and Preziosi, which gives a good introduction to a lot of the ideas we're going to cover. It's a little bit out of date now. Some of the modeling methodologies they discuss are not quite up to date, but as a reference, it's useful. So I want to now give some examples of problems that have been addressed using CompuCell. These could be anything from why blood vessels sometimes invade the retina, causing blindness, to why does acetaminophen, or if you're in Europe, paracetamol, sometimes lead to liver failure. And I'm going to give you very, very quickly a series of teasers to give you some example of the variety of problems you can solve with the same modeling environment. As Giuliano said earlier, CompuCell isn't a model, it's a way of building models. And that means that you can do almost anything in terms of modeling cellular organization with CompuCell. And so you have to decide what you want to model and how, and then CompuCell provides tools for doing that. So we have Gilberto Tomas on the line. He'll give a talk about this and show you how to actually repeat these simulations. But he did some very beautiful work on cell crawling. In the case of cell crawling, you have a cell that consists of compartments. There is what's called a lamellipodium, a thin layer full of actin that's polymerizing that pulls the cell forward. There's the cytoplasm compartment of the cell, the nucleus, which is relatively stiff. And there's a model, in this case, of how the actin polymerizes and pushes the cell forward, and the mechanics of the rest of the cell. And you can build a simulation in which the cell is sitting there and not having any decision about how it's going to move. And another simulation in which the cell is initially symmetrical, but spontaneously begins to move in a given direction. If you look at the statistics of how cells crawl, it's rather complicated, and you can show that these simulations replicate the migration statistics of real cells. Now, jumping to the other extreme, that was a very detailed model. This is also a model Gilberto worked on, is a question about cancer. Cancer is fundamentally an evolutionary disease. It's a disease of cell behaviors. And the big question in tumors is not how do you get mutations in the first place. Those happen all the time. But why are particular mutations that lead to tumorigenesis selected for in the cancer microenvironment? In this case, the question is, there are things called the hallmarks of cancer, particular patterns of behavior that are seen again and again, cancer to cancer. And the question is, if mutations are random, why do you always see the same sequence of mutations occurring or being selected for? And the key hypothesis is that the tumor microenvironment itself is selecting for certain cell behaviors. There's some beautiful work that was done quite a while ago by Heiko Enderling that showed that if you simulate tumor stem cells and tumor somatic cells in an environment with limited resources, that could be nutrient limited or space limited, you'll find that the fraction of stem cells gradually increases as the tumor grows. And in particular, if you then remove part of the tumor, lifting the limitation on resources, you see a huge jump in the fraction of stem cells. That jump in the fraction of stem cells represents an increase in cancer invasiveness. And the result of that is a paradox, which is seen, unfortunately, clinically, that when you resect a tumor, cut out part of it, kill it off using chemicals like chemotherapies or with radiation, very often, if you don't get all of the tumor, what comes back is more aggressive and more lethal than what you started with. And so very often, cancer treatments actually leave you worse off than if you'd been untreated. Gilberto and his collaborators were interested in selection for particular aspects of cell behavior. In particular, they were modeling questions of selection of cell adhesion, cell proliferation, cell movement. And what they were able to show was that even in a very simple environment with steady nutrient source, you select for less adhesive cells that are more migratory. If they added a model of immune cells, which control the tumor, if those immune response is weak, you see the same evolution to invasive behavior. I'll let this run for a minute so that it actually shows the invasive behavior. And if the immune response is strong, you see that the tumor stays compact. So the same selection pressure within the tumor, but a different immune response leads to a different outcome for the overall selection. And so the basic result in this simulation is that the nutrient gradients in the environment lead to decreasing cell-cell adhesion, increasing cell extracellular matrix adhesion, and increasing invasiveness. But in the presence of an immune system that's strong, you veto that selection, or at least slow it down, and you can delay the formation of invasive tumor. That was a very abstract example. We ran already the vasculogenesis simulation. The key question with vasculogenesis is you have a single cell type. How do you organize the complex patterns that occur in blood vessels, especially capillaries? One of the things that's interesting about vascular development is that there are two mechanisms for vascular development, one of which starts from pre-existing vasculature, that's called angiogenesis, and one of which starts from widely separated cells, it's called vasculogenesis. And the question was, this is now old work, was there a single mechanism that could explain both of these? Because the same cells are doing both. The hypothesis that we worked with a long time ago was that there was a signal, a vascular endothelial growth factor signal, which you've seen, and that chemical diffusive signal, which attracts cells, was going to be the primary mechanism for vascular patterning. In this case, we were able to show that by itself, endothelial cells subject to chemotaxis, that is chemical attraction due to a chemical they secreted themselves, were not sufficient to generate vascular patterns, so they're blobs instead. But with certain additional assumptions about either cell elongation, contact inhibition of chemotaxis, or particular mechanical properties, we were able to replicate experimentally observed work on vascular patterning in human HUVEC, human umbilical vein endothelial cells. We can now combine those two kinds of simulations of vasculature and cancer cells and ask the question, which you've seen before in the simulation, how does the presence or absence of neovascularization affect tumor growth? This again is a fairly old simulation, but here we have a growing tumor. That tumor will generate what's called a necrotic core, where the cells in the middle of the tumor don't have enough oxygen and glucose to survive. The vasculature is providing a source of oxygen and glucose. In the left-hand image, the blood vessels are proliferating in response to a signal sent by the tumor. In the right-hand side, the blood vessels don't proliferate. In both cases, the tumor keeps growing. It also moves a little bit because of differential growth. And we can ask the question, what is the pattern of growth in these two cases? And what we find is that early on in growth, there's almost no difference. Later on, we see distinct phases of growth, and those phases of growth are different depending on whether we have neoangiogenesis, that is stimulated blood vessel growth, or not. And so that's a simulation we could certainly come back to if people are interested. In developmental biology, one of the key processes, which you've also seen in the movie already, was the formation of the somites. That's the segmental structure that's going to lead to the vertebra in a chicken or human. That involves both chemical signaling, the generated periodic pattern, and mechanical simulation, signaling and movement to reorganize the cells into somites. And we've spent many years trying to understand how the interaction of chemical and mechanical signals works in this case. There's a very complex set of chemical signals within and between the cells that lead to what's called a Semitic clock. That Semitic clock interacts with cell differentiation to change the mechanical properties of the cells and allow them to organize into periodic condensed regions called somites. And that's on the left. However, there were some experiments that were done by our collaborators in England, Claudio Stern and his group, that showed that you could turn off the Semitic clock, turn off the chemical oscillators, and still get segmentation. And Giulio Belmonte, who was a graduate student at that time and who'll be lecturing in this class, was able to show that there were essentially purely mechanical hypotheses that could also give rise to segmentation. Priam who just finished her PhD, has also worked on this problem, and will teach you a little bit about how to model segmentation later in the class. Moving from these developmental problems and these rather abstract problems to much more clinical problems, we've worked on a variety of issues related to neovascularization in the retina. Age-related macular degeneration can lead to invasion of the retina by blood vessels. It's called choroidal neovascularization. And one of the questions that we were interested in is this can occur in very different ways depending on age, risk factors, and so on. We'd like to understand why you get these different patterns of blood vessel invasion and what determines risk. The retina has a relatively complicated structure. In this case, one of the things like cancer that's interesting is that you can live 75 years with no blood vessel engagement. And then within 6 months, you can lose your vision due to proliferating blood vessels in the retina. So it's a disease that starts slowly and then progresses rapidly, like some cancers. Historically, people have identified vascular endothelial growth factor A, that growth factor we studied in the angiogenesis simulation, as the primary culprit. However, drugs to treat this disease by blocking VEGF-A, which are very available because they're used in cancer treatment, things like angiostatin, don't seem to work very well. And so we built a mechanical model of what's happening in the retina, along with chemical models of what's happening in the retina. And we were able to then reproduce the pattern of blood vessel invasion and retinal damage that we see in the experiments in the clinic. And I'll let this run for a second. While it's running, I want to call out the fact that we have blood vessels. The blood vessels are connected to each other. They stick to a layer called Brooks membrane, which is the organizing layer at the back of the retina. The black cells in your eye, the pigmented retinal cells, stick strongly to that Brooks membrane. Above those pigmented retinal cells, there are photoreceptors, which stick a little bit less strongly. And depending on what happens to disrupt these various aspects of adhesion, you can get different risks of blood vessel invasion. And so in this particular cube, if we have lipid accumulation with aging, that is classical risk factor for age-related macular degeneration, that reduces the adhesion between the pigmented epithelial cells and the Brooks membrane, and that increases the risk of invasion of blood vessels between the membrane and the pigmented retinal cells. Other kinds of damage to the retina can cause other kinds of risk and other kinds of invasion patterns. And here, the simulation is completed after a year with the breakup of the retina, which would lead to blindness. This seemed to be a stretch when we did it quite a while ago, but in fact, some years later, there were experimental works done that showed that there were, in fact, adhesion defects, in particular adhesion between pigmented retinal epithelium and Brooks membrane present in people who had this disease. Another disease we worked on, which had more clinical outcome, was polycystic kidney disease. This is a disease in which cysts form in the kidneys, and unfortunately, it's untreatable without a kidney transplant. People's kidneys get up to the size of watermelons sometimes before they die. It's a rare disease in the sense that it's not something that probably any of you will get, fortunately, but it's not that rare. It's about 1 in 1,000 incidents. It's a genetic disease, which is fairly unusual. Mutations in 2 genes essentially deterministically cause this disease. If you have those mutations, you'll get it. If you don't have those mutations, you're extremely unlikely to get it. However, we didn't know how those mutations in that gene affected the phenotype of cyst formation. There was an observation that there was a particular cadherin, a particular cell adhesion molecule, that was inappropriately expressed in people with this disease. Interestingly, that was what's called a juvenile cadherin. So this is a molecule that's present in normal kidneys while they're developing and growing, but it should be turned off in adults. It's not turned off completely in people who get the disease. Cadherins are molecules that help cells stick together, but they also send signals. And so a cadherin defect could be causing a problem in a variety of ways. In these simulations, we tried to analyze cyst formation, subject to a variety of different hypotheses about what this cadherin, this extra cadherin, could be doing mechanistically to the cell behaviors. We were able to show that some of these hypotheses agreed with the in vitro experiments that we did. Others didn't. In particular, a hypothesis that the cadherins were actually interfering with adhesion rather than with cell signaling agreed better with what we were seeing experimentally. In this case, we went back to more classical bioinformatics approaches to analyze what that meant in terms of the significance. And we were able to identify some drug targets that potentially could be used to develop treatments for the disease. Those drug targets work in vitro. They seem to work in mice. Of course, curing mice is easy compared to curing humans. But our clinical collaborators are trying to tool up to do a clinical trial for some of these drugs. Diabetic retinopathy is another vascular disease in the retina, this time involving blood vessels in the front of the retina rather than the back. And in this case, instead of getting too many blood vessels, you're losing blood vessels. It's treated currently either by injection of anti-VEGFs, the same molecules we were talking about earlier, or by actually going in with a laser and ablating, that is burning out parts of the retina. And this laser ablation therapy is pretty violent. Here on the lower right, you actually see holes in the retina provided by laser burns. At the moment, there's no real way to target those holes so that they damage as little bit of the retina as possible. Every one of those holes is a place your photoreceptors don't exist. So this is a treatment that's essentially cutting holes in your retina and reducing your vision. Not a very pleasant way of treating. It's a little bit like fire breaking when you have forest fire. You cut trees down to prevent the fire from spreading. But in this case, they're essentially cutting the trees down randomly instead of cutting them down where you think the fire is going to spread. And so our goal is to come up with a computation that would allow us to predict where the fire will spread so that you could cut out the trees, that is the photoreceptors, only where you needed to. There was a basic hypothesis about how oxygen provided by the blood vessels interacted with vascular growth factors, and how those vascular growth factors in turn led to failure of blood vessels. Insufficient growth factors can cause blood vessel death, but excessive levels of growth factors could also cause blood vessel death. So in this case, you're getting blood vessel exposed to levels of growth factors that are way too high, and that's causing damage. That also happens in cancer, by the way. It's a very similar phenomenon. We were starting from images of the retina that were taken by our collaborators in the optometry school here. And so we digitized little patches of retina with the blood vessels from individuals. And then we built a simulation of blood transport and oxygenation in the retina and the release of vascular growth factors. In this case, we start from a simulation in which there's no excess vascular growth factor. And then we ask the question, if we have a blood vessel disappear and some level of blood vessel disappearance is normal as you age, what's the risk that the disappearance of one blood vessel will lead to a cascade of damage throughout the retina? And you can then assign a risk factor, just the way we did for that choroidal neovascularization. A risk factor for each blood vessel, saying if this blood vessel disappears, it has a high probability of causing a problem or low probability. If it has a high probability, then you want to ablate the retina near it. If it has a low probability, you want to leave it alone and save the retina. And so we were then able to do a calculation that said, if you ablated particular regions, what's the area of damage directly from the treatment? And what's the probability that you're going to have damage later if you from the untreated regions? And you're then able to come up with an optimum, which trades off the cost of the treatment, which is damage to the retina directly, from the benefit of the treatment, which is reduced risk of spread from the disease. The last one I want to talk about was liver toxicity. I mentioned acetaminophen as a liver toxin. One of the issues with hepatotoxicity is it's very idiosyncratic. One person could take a drug perfectly safely, another person might die from it. Even the same person might be fine at taking the same amount of the drug. One day might be fine, another day might have a problem. And so we were very interested in trying to understand some of the origins of that variation. Tylenol, acetaminophen, is very toxic for the liver. There have been lawsuits for decades over it. In many cases, it's used safely, but especially when it's taken after alcohol consumption, can be lethal. And so one of the questions is, what is going on and why? And this is a bit surprising because if I were to surgically remove 75% of your liver would grow back in a few weeks. You wouldn't even know that it had been done. In the case of acetaminophen, the direct insult to the liver from the poison only kills about 15–20% of the cells in the liver, and yet your liver goes on to fail completely. And that's a bit mysterious. Why is the liver not able to recover from an acetaminophen insult? It's able to recover from a surgical insult. And so we built a series of simulations of the transport of acetaminophen in the blood, its movement into hepatocytes, into the cells in the liver, its metabolism, because actually acetaminophen itself isn't toxic. What's toxic is one of its metabolic byproducts, coupled with its metabolic byproducts, and then the cell damage due to that byproduct. And in this case, we're able to calibrate that model so that the time course of the concentration of acetaminophen and its byproducts in the blood matches that's observed clinically. Once you have a model of that kind, you can change the parameters in the model and ask, how does the damage change? That is a sensitivity analysis of the model in terms of the parameters. And you can then build a simulated population of individuals with different parameters representing different potential genetic markers for the liver, and ask the question, are there rare individuals who are either very resistant or very susceptible to APAP damage? The answer is that even fairly small changes in the parameters, if you have the wrong pattern of changes, can lead to big changes in susceptibility. So this suggests that, in fact, that idiosyncratic response that you see clinically really can happen even from a very simple model. So I'd like to take a few minutes now to do a simple exercise, maybe not so simple, to start you on an exercise. It's the only exercise we're going to have in this module. I'd like you to take just 5 minutes to 10 minutes to think about modeling strategies. And maybe if you're going to join the hackathon, think about what you'd like to do there. And so I'd like you to think about a biological or biomedical problem you might want to work on. Presumably, if you're taking this workshop, you have some problem of interest. If you're a student, there might still be something that particularly interests you. And then what are the key questions you want to answer? What are the biological hypotheses you want to test? Think a little bit about whether a multicellular model is the appropriate thing to use. Should you be using a continuum model? Should you be using a molecular model? Should you be using something else? Ask that question. And then think about what experimental data might be available to build the model or to validate it or to test it. Do we know enough about the science to build a model of the problem you're interested in? If you did build a model, how might you compare the simulation to the experiment? That's always a difficult problem. You might want to think about that. And then if you had a validated model, what would you do with it? What good would it be? What would you learn from it? How would you use it? Would you be designing a surgical implant? Would you be designing an engineered tissue? Would you be designing a drug therapy? What would you use it for? And then if you did use it, are there experiments that you could design using that model? because one of the most powerful things you can get out of this kind of modeling is predictions that you can then test in experiment. Running simulations is cheap. Doing animal experiments and human experiments is expensive and difficult. But if you can focus the experiments that you want to do, that's very valuable. And then if you were going to do those experiments, what could you manipulate? If it's a human, probably not much, unless it's a drug trial, maybe surgical. In an animal, even, you also have limited ability to manipulate the biology. What could you do? So I'd like you to just take a few minutes to think about those things. And then when you go home, or since you're at home, when you're offline, maybe think about fleshing out these ideas. Once you've made a preliminary idea about what you want to do, think about what the key biological components of a model might be. What kinds of cells would you have? What kinds of extracellular matrix would you have? What organization would you have? What scales? What are the mechanisms? What are the cells doing? What's the tissue doing during the phenomenon you're interested in? How would you set it up? Would you be able to get measurements from an individual patient? Would you be getting measurements out of some microscopy experiment? Would you be making up the configuration? Would the configuration arise spontaneously? And since you're going to only be modeling a small volume of tissue, probably a cubic millimeter, a couple of cubic millimeters, you have to think about what happens outside the region in which you're simulating. What happens over time? So those are all kinds of questions that you might want to address in a simulation of this kind. And so I'd like everybody to take just a few minutes. These problem sets will be available online. Not all of them are yet, but they will be. And I'd like everybody to take a few minutes to think about this in their context. And if there are any questions that have been asked in the Slack or the chat, I'll try to answer them while you're working on this. So let's just take, so already we're running a little bit behind, it's 1:20. Let's say we'll take till just 1:30 and let each person work on this on their own. If you create a document of this kind and you want to share it with people, with us or with others, that would be great. If you're going to participate in the hackathon, this is a very important document to have because it would help you define the problem you might want to work on in the hackathon. So I hope that that was helpful. And that's going to be the end of this slide deck, I think. And I want you to give you, again, just a few minutes to work on this and think about it before we move on. Okay, let me go back and look at some of the questions that were asked. So there's a question about measuring integrin and cadherin in the example. I think you were talking in this case about the cancer simulation. So in the cancer simulation that I showed of cancer evolution, there was no explicit modeling of, no explicit experiment which we were anchoring to. However, it is known from many studies of solid tumor somatic evolution that in general, cadherin expression, in particular E-cadherins, so cadherins associated with epithelial layers, are downregulated during the progression of cancer. And integrins are often upregulated. Integrins help cells stick to the extracellular matrix. And so that was, again, a very abstract model. It wasn't one that was a model of a particular cancer type, although it would be very interesting to be able to do that. Okay, there was another question. Can CompuCell be adapted to simulate behavior of multiple solid organs with signaling and cell migration between organs via blood vessels? Well, the answer is, in principle, yes. The big question when you do a model of that kind is how many cells you're going to simulate. The simulations that we're able to do with CompuCell typically max out at about 50,000 to 100,000 cells. And so you're limited to fairly small volumes. If you want to have multiple regions, so for example, two 1 millimeter cubed regions that are connected by a vascular transport model, you can do that. And we're going to show you later about how to connect. In fact, TJ in his next talk will talk about simulating the transport of cells and components in and out of tissues. That liver model that I showed had a PBPK model of transport and metabolism in the whole body, coupled to that detailed model of a small fraction of the liver. So in principle, you can definitely do models of that kind. They tend to be computationally expensive. And so the question then is, what are the specific questions you want to address? But in principle, yes, you can certainly do that. If you want to actually model the mechanics of cells moving, for example, through a blood vessel or a lymph node, CompuCell probably isn't the best place to do that. The models like that one I showed of Carniodacus modeling the individual blood cells in great mechanical detail would be more appropriate. Again, those are very expensive models to run, but they're also very beautiful and productive. What is my approach to gauging if there's enough experimental data and prioritizing experiments? Well, one of the things that is really important is actually doing the exercise that I'm just laying out. If you can identify what the components of the model have to be and what their interactions and behaviors are, and there are at least some experimental data or literature knowledge that allow you to begin to constrain the possibility of behaviors and interactions, then you can begin to address, build a simulation. If they're missing pieces, and very often you find out that there are, gee, we don't know what happens when cell A touches cell B, then you have to either have some hypotheses about what those possibilities are, in which case those would be experiments you want to go back and do, or you may find that there just isn't enough information to build. But that's a very good question, and that's something that is very often the process of building the model is very informative, even if you actually never run it. And so I think that's quite a good question. There's a question, how is cellular heterogeneity taken into consideration in these models? We introduce random noise. So there are a bunch of different kinds of heterogeneity and noise that come up in biology. The way cells move is noisy. The way molecular signals happen is very noisy. Both of those are typically included in models. Actually, the noise in chemical signaling, less. The spatial noise is included explicitly in CompuCell. Stochastic movement of cells is included. And we'll see in the next talk that stochasticity shows up in a big way in the simulations. And you can also ask the question, for example, about differences of gene expression. If two cells of the same type are both expressing, say, cadherin, E-cadherin, the level of E-cadherin and its distribution in the cell will be different. And so we'll show you how to build simulations in which the amount of particular chemicals or their spatial distribution or the individual parameters of the cell, for example, how motile it is, differ between individual cells. That tumor simulation of stochastic evolution was a good example of how to do that. Maybe Gilberto could show people a little bit about how to do that later in the class. Okay. Can two biological pathways be meshed up in one model? Absolutely. If you took Herbert's class, combining pathway models is a very important part of that. We'll do a little bit of that in this class. There isn't a universal way to do it because it depends a little bit on how the models are written and described. but very often the same molecule will show up being used in different ways in different pathways. And so that means you have crosstalk between the pathways, and you have to consider that when you build a simulation. As an example, the particular way in which liver cells are damaged by APAP, by Tylenol, is through depletion of a molecule called glutathione. Glutathione is a buffer for a reactive oxygen species. So if you have depletion of glutathione, you have cell damage due to reactive oxygen species that can lead to cell death due to mitochondrial permeabilization, DNA damage, a variety of other ROS death mechanisms. It winds up that immune cells, T cells in particular, kill cells by ROS mechanisms. And so if you have very high levels of glutathione, cells are somewhat resistant to being killed by T cells. One of the reasons that, for example, pancreatic cancer is so invasive and so destructive is that cytotoxic chemotherapies and T cells both have a hard time of killing pancreatic cells. Pancreatic cells are intrinsically in a high reactive oxygen species environment. They have high levels of glutathione to begin with, makes them hard to kill. The cancers tend to upregulate glutathione production even more, making them even harder to kill. And so that would be an example where the same signaling pathway and same molecule, so in this case, a metabolic rather than signaling pathway, same molecule, glutathione, which is playing a protective function in the liver, in the case of cancer, is making the cancer more deadly. So again, yes, somebody's saying using particular simulation tools to do integration of network models. We're going to use the simple tool Tellurium that Herbert Sauro taught last week. As a tool for learning, I find that the easiest and fastest way to get up to speed on network modeling. However, there are a variety of very sophisticated tools that are designed for parameter fitting and for model integration. Copasi is one, Cell Designer is another. And so just the way there are many tools for doing multi-scale, multicellular models, there are many tools available also for doing individual network models and for integrating them. Well, that was the end of the time we had allocated. Again, this is an exercise which you can carry on at home. Was anybody at least able to write down a few ideas about a problem they're interested in and potentially a strategy for addressing the problem? Again, the hardest part and the most important one is to identify what the problems are that you want to solve. And the more specific and quantitative you can be about what those hypotheses would require to be tested, the more likely you are to be successful. Somebody asks, how does CC3D compare with Morpheus? What are the strengths and weaknesses of each? The answer is we work very closely with the people in Dresden. We visit them periodically, at least we did until the disaster. And we work with them also on the COVID modeling. Morpheus has a beautiful model specification framework. It is, however, a custom model specification framework that's unique to Morpheus. CompuCell uses Python, and so you don't have to learn a new language if you already know Python. I'd say that they're pretty comparable in terms of capabilities. It's really more of a matter of taste. There are aspects of model design that I think are wonderful in Morpheus. I wish we had them in CompuCell. There's some other places where Morpheus doesn't have, for example, as many mechanisms. For example, we have a concept of solid links, junctional adhesion between cells that Morpheus doesn't have. And so anything you learn using CompuCell, you can take over and in a day apply to a Morpheus model and vice versa. In fact, about a year ago, Giuliano and I and some other people went over to Dresden to a big meeting on the subject. Giuliano was writing code to try to translate Morpheus models into CompuCell models and back and forth. You can't quite do it automatically, but it's pretty close. After all, if you're modeling the same biology, you should get the same results. The modeling framework shouldn't matter. So that's a great question. Okay. All right. So that was the end of that slide deck. If people want to take a break for a minute or two, why don't we take a 2-minute break, and then we will tool up. We'll tool up for TJ talking about COVID modeling. So TJ, are you there?
Yes, I'm here, James.
Were we supposed to, let's see, it's 1:30, that's 2.5 hours. Were we supposed to have our lunch break now or after your first talk?